Thesis on computational biology. This is the progression towards modeling of cancer cells within the LAMPS molecular dynamics software. So, I should probably actually do this. Crap. I'm just gonna cut that out. So, good afternoon. This is my thesis on the computational biology. It's about the progression of towards the modeling of cancer cells within the LAMPS molecular dynamics software. So the ultimate goal of my research is to study the migration of cancer cells within healthy tissue by simulating these systems of normal cells. To do this, a bunch of intermediate milestones will have to be met first. First, a simulation method will have to be developed, then developing a normal cell and validating it, developing the cancer cell and validating it, and then developing this interaction or migration simulation, which I've called the metastasis simulation. Currently, the simulation method has been developed, and my current work is in developing the normal cell model. I've currently done the geometry work for this, and the parts of the membrane, cytoskeleton, internal and external fluid, and nucleus have been modeled. Currently, development is in the simulation code to run this geometry file. The graphic on the left shows behavior, which is shown in Fu et al. 2017. This is the kind of behavior we're kind of targeting just because it's very cell-like and it's able to be validated quite easily. There's a lot of resources out there validating red blood cell behavior like this. This is also one of my main references in this research, this Fu et al. So an overview of LAMPS, which is my main tool. It's a molecular dynamics code. It has a focus on materials modeling, but is extended into a lot of different materials such as metals, semiconductors, biomolecules, polymers, and coarse-grained mesoscopic systems. It's open source and openly distributed, so it's pretty easy to customize code how you wish and modify code as you see fit. LAMPS also has a very active mailing list with users and developers of the code, which can be a great resource with for information and problems that people might run into while developing. So this is a sample of my LAMPS workflow. So first I'll have an idea for a simulation or the goal simulation in mind. And to do this, technically you can run straight from the LAMPS input file. The problem from this is that the geometry creation options within the LAMPS input files are quite limited, and creating complex geometries is virtually impossible in these. So it's much easier to follow a LAMPS data file format, which they're open about, and use a Python script or some other scripting software to write out this file without calculating these by hand, because this would take a very long time to do all this. So once the data file and the input file are written, the data file contains the initial conditions and the initial positions of all the atoms, and the LAMPS input file sets the settings for the simulation and the behaviors which each of these particles will follow. And then once these are all created, they're fed into LAMPS, which is a Linux-based software. So I use an Ubuntu shell operating system to run this. LAMPS then uses or produces dump files and can also produce raw data files to text files. For the dump files, I use in programs such as Ovito, which is an image processor, and it allows me to produce images and videos of these simulations, and can also do some other data analysis as well within Ovito. So building data files within Python is done for me, at least, because Python is a very intuitive and user-friendly programming language. It's not necessary at all to use Python, but I just find it easier to write them this way. Other people have used MATLAB, like the Fu paper uses MATLAB to write their geometries. But essentially how this is done is I set specific parameters and geometrical algorithms that are used to calculate atom positions and their properties. And then I use different, I create an array using these parameters and these algorithms and go through each point in the array, which represents the simulation space. And the simulation space is then iterated through to decide which points will become which types of particles. So running the simulation with Ubuntu, as I said, it'll produce the dump files. And this is a screenshot of what it looks like running something within Ubuntu. And then this is what it looks like putting a dump file into Ovito. As you can see, you have these different views and such, but the dump files can be processed into images as these stills you've seen right here, or they can process into videos at varying resolutions and other modifications can be done for these as well, such as you can construct meshes across different sets of points, displacement vectors for individual particles. You can represent atoms by different colors and different shapes and such. And there's a lot of other things that can be done as well. This is a pretty involved program. I've only scratched the surface on some of the stuff it can do. So then also recently I've been running into some problems with some unknown errors in lamps. It's a certain kind of error that comes up when there's an unresolved error within lamps, and external tools were needed to debug this. These tools were used to create a stack trace, which program developers can use to identify where the problem is a bit more. And this helped me identify the source of the problem being an atom style that was being used. It was far too complicated. Also, the hardware of simulations is important to go over as well, because running a simulation on a single core can take hours and hours and hours, whereas if effective hardware acceleration is done, this can take a matter of seconds. So there's multiple inbuilt functions in lamps for accelerating simulations. This can be done using CPU, GPU. There's cluster computer options and other things as well. Currently, the only acceleration I've used is CPU acceleration, because I have a multi-threaded CPU personally, so I'm able to speed up simulations fairly well this way. But GPU acceleration would give a significant speed up over that even, I believe. So that would be a point to develop later. So currently my main work is taking the geometry you see on the right, which is the geometry I created, and getting it to try to act like the behavior shown before on the left. The input file for the simulation, or the input file for the geometry on the right, has been made and it's stable, but the behavior is clearly not the same. So I've been using the reference of Foo 2017 to try to develop my input file and my simulation in general to more fit this behavior. Excuse me. So to create this cell model, obviously I used Python, like I said, and I kept constants. I set a couple of constants in particular to make sure that the diameter of the cell is 8 microns. And again, using scripting, went through the spaces and array and placed all the water, membrane, cytoskeleton, and such. And then this is the video of the simulation I have. It's kind of hard to see exactly what's happening, honestly, just because there's not much interactions. It's mostly just translating to the left very slowly, and all the particles are kind of staying in the same configuration. It's not the worst reaction. It's not blowing up, so to speak, which is something that's happened a lot in some of my simulations, but it's clearly not also the correct behavior. So I think I know why this is partially. It's partially due to only using a simple NVE fix, and I just wanted to make sure I could run this geometry with a simple simulation with no coding mistakes. So this is just kind of more of a prototype more than anything. And this is just another video where I shrunk down the water so it's a little bit easier to see. So then this is that Fu et al. Paper. Their full name of the paper is Leonard Jones Type Pair Potential Method for Coarse-Grained Lipid Bilayer Membrane Simulations in Lamps. So their research was into kind of doing the same thing I'm doing, except they kind of stopped at the point where they developed the cell. I want to continue past there. So the reference of the paper has been helpful for doing stuff as well, just because they explain their methods and such. They also provided their files, such as their data files, their input files. But as you can see, this paper came out in 2017, so it's a few years old now. And due to that, LAMPS has been updated a number of times since then. And this is a problem with a couple of other files as well. It seems that the updates have made them not work or something. So I've tried to replicate the behavior shown in some of these pictures, and the geometry shown I have replicated. That's from their paper, but the behavior is something that still eludes me. But I still wish to. I've tried to fix their files to get this behavior, and I have had some success. As you can see from these pictures, I was able to clean up some of their files and fix up the MATLAB file, which they used to create their geometry in particular, and I'm able to create the geometry they had. The lamps input file is a little bit more problematic in particular, which I'll get into a little bit later. But I was able to make it work enough to run a simulation, but it's not quite what they had in particular. So The simulation has two main steps. It has an energy minimization step, which allows particles to get to a lower energy state before the simulation start. This kind of helps with simulations not blowing up. And then the simulation step after the minimization step is there's a fix imposed on the internal water to change its volume, as was shown in this slide. The V corresponds to that change in volume, and that's used to control some of the behaviors of the cell. So that simulation step is the second part you will be seeing in these coming videos. And you'll also see that these behaviors clearly don't match. So there's a couple moderate changes I made to the input file to make them to be able to run. The biggest change I made in particular was they had some custom potentials for their membrane, which were written in C++, and I tried to unbug, but these were pretty bugged, and I was unable to fix these up to get them working. So I'm using different, fairly similar potentials, but as you can see, this is probably the source of the problems. So this is my first video. You can see at the beginning that during this energy minimization step, just the water is moving around and the membrane stays nice and still. But once all of the interactions are fixed in towards a quarter of the way through, you can see the membrane kind of dissolves and doesn't keep its shape. Yeah. But this is still in advance over some of my other simulations, and I did some modification to some of the visuals so we can see a little bit better. This is without the water particles. I'm going to skip this one because it's actually better if we just go right to with the membrane particles as well shrunk down. So we can actually see the behavior of the cytoskeleton and internal water acting fairly correctly actually before that membrane kind of lets it all go. So this is progress, but it's not perfect yet. And you can kind of see the pink particles as well kind of stay clumped together more correctly. So it's part of the future work that's going to need to be done. This section's going into that. First are my ideas for attaining the proper behaviors, I would say, which is kind of like what they get in Fu et al. 2017. So I would say the first thing to look into would be those custom pair potentials by Fu. This seems to be the biggest change, and this was the biggest change I made only on the membrane, which seems to be what's not acting correctly, so to speak. I'm not a huge expert in C++, but it seems that these problems are related to updates in lamps. So if these can't be fixed or salvaged, so to speak, these new potentials are going to have to be developed or found. There's also a couple different geometrical patterns and such in Fu that are used that I could implement into my geometry as well that might help as well, such as anchor points between the membrane and cytoskeleton, which help them kind of stay together a bit better. And then after that, the next thing's going to need to be done is validating this cell plan. Fu also validates their plan or validates their cell within their paper by using this other paper from 2014, which I show here, erythrocyte response. In this paper, they use a numerical model of a red blood cell, and they validate it, comparing it to real-world data to make sure that the shape and curvature of the blood cell are comporting to reality under specific conditions. That was kind of what I was showing with those changes in volume during the Fu slide. That's what they were doing to validate their behavior. So once the normal cell is developed, this same method should be applicable to validating the model because we're going to try to, or because the method which is being developed is fairly similar to the Fu method. And then other options for validations also exist. There's a paper called a parallel fluid solid coupling model using lamps and polybos based on the immersed boundary method. This also offers a method which could be used to validate the simulation. There's also a lot of other papers out there which could be used to validate cell behavior when applied to this research. After this, the cell or cancer cell will have to be developed and validated as well. There's also a few papers as well which I've found out that investigate mechanical and chemical differences between cancer and normal cells. The physical differences are obviously are going to be more important than the chemical ones in this case. So to get this model of a cancer cell, the simplest method is most likely going to be to take the normal cell template and then modify structure or behavior around this already validated cell to represent the cancer cells. And then I also have a couple of papers linked here to help with some of the changes which might be made for these cancer cells, as well as the validation. There's data within this 2008 paper which might be used for validation of this cancer cell. Then lastly, a metastasis simulation will be made. So once we get both these types of cells made and validated, then the simulation where put them all together, like get big groups of them together and see how the cancer cells will migrate through the normal cells uh, will be at this point. This is gonna be a huge simulation most likely because if it's containing even 100 cells, each cell is made up of approximately 20,000 particles. So on top of developing all this behavior as well, one thing that'll be important is developing some hardware acceleration, just because running one of these simulations is likely going to take a very, very, very long time unless we find some way to accelerate it. And all the interactions will need to be tested as well and validated from normal to normal cells, cancer to normal cells, and cancer to cancer cells before we can ensure that this is good data, essentially. So then there's a couple improvements, which are smaller things, which are less impactful in the final product, but might be helpful for developing towards that final goal. A Python and LAMMPS integration would be helpful because LAMMPS has been developing functionality where they can implement their code as a library within Python. This would be really helpful because it would significantly streamline the workflow where I wouldn't have to develop a separate input file and data file. This could all be done within one file, and LAMMPS could possibly be even invoked within Python. So this could make my workflow go from what was shown to essentially Python to a veto to the data. It would be much easier. A smoother membrane might also be easier. The current algorithms are a little bit rough, meaning the iteration limits are kind of high, so the spaces between each spot that's checked is kind of large. And if these are shrunk down, it could give a more continuous and smooth surface for the membrane in particular. And it could also help with placing more atoms accurately in general. There's also Palabos integration, which is a code that was linked in one of the references that or models water at a much faster rate than LAMMPS. This could help with a more accurate and faster water model. This could take a lot of work though because it is a separate code and the coupling is pretty intricate. More subcellular organelles and cell parts would also be helpful as well to more accurately model the membrane. A better cytoskeleton where it's kind of more distributed through the cell would probably be better, as well as anchor points and surface proteins on my geometry. And then again, simulation acceleration is important as well with GPU, cluster computing, and then also if I could divide up the tasks for each computing core within the simulation better, that could lead to an acceleration. There's also a set of research called the ELBA water and lipid methods, I would say. These were a set of papers that were written a number of years ago by a couple of researchers. They went into self-assembling lipid molecules and water models. If the Palabos integration isn't used, this is a very good way to potentially model water as at a less costly way with a more accurate model. However, the lipid side of this and the membrane side of this is a little bit different than what we're shooting for, so I don't think it will be super helpful. And then I'm just going to go over a couple of my my progress or my progression steps in simulating in LAMMPS. Some of the names of these videos are weird too if you look at them on YouTube. This is the tip of the iceberg of what I've created. So there's a lot of random names I've made just because like trying to come up with random names gets kind of hard after a while actually. So there we go. So this is my first simulation progress video. Some of you may have seen this one already, but this goes from approximately August of this year until, or goes up until August of this year, I think. I think that's when it was. Yeah, a lot of these particles are made up of very massive numbers of particles, actually. And these are all rigid bodies as well, so they're not interacting at all with each other, actually. They're just completely rigid. These ones were not completely rigid, but obviously not acting like real world either. And these ones are not rigid, but as you can see, they don't really hold together at all either. So at that point, it was kind of an all or nothing with holding together. And then this was the start of the exploding simulations. What I found out is eventually some of these behaviors happen because of excessive energy within the system. It's kind of why I went over the minimization step earlier. Those kind of steps can help eliminate some of these behaviors. And this was trying to create a watertight membrane, and it wasn't quite successful, as you can see. And that's not true. This is definitely not my most current sim anymore. So yeah, that was my progress up until about August of this year, I believe. Um, so then my next one was I had this, which I'm calling the soft ball simulation. This was kind of similar, except this is when we were thinking of modeling the cell as a collection of individual points that was solid. And as you can see, this didn't really model a cell very well. So this method was kind of shelved for the moment. This is when I was getting better at placing atoms in just a shell, but as you can see, the atoms kind of disappear immediately. This turned out to be a bug where in a geometry or like creation, it was placing atoms directly on top of one another, thus causing them to kind of bug out and disappear immediately in the simulation. This is another view of that same kind of thing. You can see just kind of poof out of existence. So then this is my first stable simulation. This one's kind of similar to uh, the my geometry membrane I show, where it's just moving very slowly to the side, and it has mostly interactions with just the velocity, but there are some interparticle interactions as well. They're just not very strong because I learned to reduce the amount of energy within the system. So then again, here are some of the pictures of the geometry which I have made. You can see on the top, it's just one layer of water, whereas the green is the external water. Whereas on the bottom, it's kind of filled more so. Then the blue is supposed to be kind of a cytoskeleton model, and the yellow is the nucleus, and the pink is the internal fluid or water. Then here's the videos. Again, I already showed these, so we can just kind of go over these quickly. And then these were some other geometrical models I had produced a little while ago. These were kind of another method we were thinking of, which I was calling the sparse membrane method. This would have used fewer particles spaced out more evenly to using much longer longer range interactions. This could work as well. It's just a different method for creating a membrane. This geometry was created using a Fibonacci sequence to evenly space out these points. It's kind of hard to see the exact structure, but they're kind of spirals upwards. And this pattern could also be used in my other geometry or creations to possibly place other points, such as like those anchor points or other things throughout an existing membrane. And I already showed you the fluid membrane modified. So yeah, those are my references. And thank you for listening. If you have any questions, ask me in just a moment. There's my email, not connected to UWM, if you have any further questions. And thank you very much for listening.